We have been talking about the development of the brain, and if you look back in the last 3 or 4 lectures, we have come a long way from trying to understand how the cells started from the germinal layer to the development of the various brain parts, and whether there was a program, genetic program, nature devised it, and cells follow it accordingly, or whether there were some external influences. How do cells proliferate? How do cells aggregate? By now you do understand these terms and do understand what stands behind these terms. A whole series of actions: cell death, survival of the fittest, cell migration, cell aggregation, formation of nuclei, and so on and so forth. And then we talked about the 3 major hypotheses: how cells aggregate into nuclei and how cells know where to travel. And we did assume that each one of these hypotheses did have support, but we are still waiting which one stands out more, and let's wait for the future to give us evidence for it. Then we moved in to the stages of growth, how the brain of the prenatal to the neonate to the postnatal periods, 3 months, 5 months, right up to 14 years. And we talked about how the brain is developing till about 14 years. Myelination is taking place, insulation of neurons, connections of neurons. And therefore, what it means is that there is a lot that is going on within the young and the adolescent brain. We also talked about whether it is possible for neurogenesis to take place in the adult brain. And yes, evidence from birds and lower order mammalian species does indicate that there is some neurogenesis taking place in the adult brain. And we also talked about the sprouting of connections, enlarging of certain areas of the brain in the adult after learning. So stimulation is extremely important. Today we are going to talk about where we ended last time in lecture 20. And this is extremely important for you to understand as young people, that there are influences in the embryonic, in the fetal, in the embryonic and the postnatal development of the young immediately after birth, which are extremely important, and they can change, alter, degenerate, or even help the brain grow. And what are these influences? We're going to talk about them, but remember, it is the presence of or the absence of needed nutrients or lessening of or increasing of the required amounts. Either way, it works to change the already developed template of the brain development. Now, we did count a few of those influences. We talked about nutrition being very important, and we are going to go into the details. Hormones, the male hormone, the female hormone, the thyroid, which is the growth hormone. Then there is the external stimulation, and we had referred to it earlier in Hubel and Wiesel's experiments. We had also referred to, but we had not really talked about oxygen. You do know oxygen is extremely important for each one of us to survive. Similarly, for each one of the cells in the brain, and similarly for each one of the locations at it. So, therefore, these are important, but also is important the motor stimulation. Unless you exercise, you remember, exercising the connections, it makes a difference. So, exercising the stimulating muscles thereby strengthens the connections within the brain. So, we will talk about what can go wrong and what can go right. Then viral and other infections, measles, meningitis, and even Some mothers have, let's say, disorders which are like they're asthmatic. Then the drugs that they use, there is caution, and we'll talk about, or antibiotics that they take, which is again refers us to drugs. Children born to women who have been taking drugs. And then radiation, exposure to radiation, X-ray, simplest, where every mother just, doctor says, you need to take an X-ray, and so she goes in. Does that make a difference? Radiation from other sources. And there are instances where we have seen devastating effects of radiation. How does, your question, how does this happen? Does the fetus not have its own protection? How can nature not develop a system by which the fetus is protected? Well, the protection is the placental barrier. But the substances that we talk about are those that, and the fetus is very well protected. But at the same time, it is at the mercy of the mother, because it's getting all its nutrients, all its supplies, its lifeline, so to speak, is the umbilical cord. And therefore, whatever is passed on through the umbilical cord will get a good passage right into the fetus and the system of the fetus. These periods are known as the critical periods or the sensitive periods. The sensitive periods or the critical periods, why are they called the critical periods? These are the periods within which certain influences. Before that, the influence will not affect the development, and after that it will not affect it. So there is a boundary, a time limit within, or a growth limit within which that particular influence will affect the rate of development or the growth or whatever is happening within the brain at that point in time. So the periods within which the growth of the organism can be affected by influences, that is the period of development, the period of differentiation, the period of proliferation, the period of forming connections, or the period of aggregating, and then receiving stimulus and stimulation from the external environment. The period prior to or subsequent to this period is not important. And even if it's like closing the barn door after the horse is stolen, will not make a difference. The horse will not come back. In the same manner, if you add substances after the critical period is over, that will not make a difference. So the influences and substances will not affect development before the periods, the critical period and the time boundary, and after the critical period or after the time boundary of that particular development, of that particular phase of development is over. So the brain is most susceptible and only during that particular limited time. And what influence and devastating influence it can have. Normal development can be affected and is affected by these influences, by the presence or the absence of, by the increases or decreases of normal levels that are required. So first of all, it is either it is present or absent. It is present when it is not required to be, or it is absent when it is required to be present. That particular substance, that particular chemical, that particular nutrient, or that particular whatever will have an effect, and that is the series, the list that I gave you earlier. Similarly, increases more than normal, which is required, or decreases from the normal level that is required by the brain to grow, will have a long-lasting and sometimes irreversible effect on the development of the brain. Now this, you must understand, in some cases the effect is irreversible. Beyond that time boundary, If you keep adding substances, it will not make a difference. Okay, let's talk about the first one, which is nutrition. Nutrition is important. We all agree. Why? Nutrition provides important resources such as proteins, amino acids, glucose, etc., etc., for the growing brain. Most importantly, proteins are very needed. And nerve growth factor is coming out of the substances. So nerves connecting, forming, the starved neurons will not be able to sprout. And where does this happen? Mostly during the early fetal periods when the brain is growing. Malnutrition during this period, where does it come from? Yes, where does this come from? Yes, it comes from the mother. If the mother is malnourished, the consequences, she is adult, she will bear the consequences because her brain is grown. The cells, if they die off, they're not going to be a devastating effect. But where is it going to have a devastating effect? And that is the fetus she is carrying. So malnutrition during the period when brain cells are dividing and multiplying rapidly. Remember the word, the magic number, 20,000 neurons per minute. Cell death will be rapid. Instead of cells sustaining themselves, they'll die off. So, first of all, what we'll see is decrease number of cells that will emerge and decrease in the division of cells. So, decreased division of cells, decreased number of cells, increased cell death. So, you're left with what? Fewer cells in the brain. Now, this period of starvation of the brain, impoverishment of the brain, is from the second semester to the first year of life. So, second semester, which means after the second or the third month, onwards to the first year post-natal. That's a long period. About 6 months in the uterus, in utero, and about a year, almost 2 years. And this is where neurons of different sections of the brain are forming, developing, connections are forming. There are different times, boundaries and time lags and time schedules for each one of the areas of the brain. The effect of malnutrition will depend on the area during which that malnutrition has occurred, upon the time in which those specific cells were dividing. So, if cells were dividing and moving to the cortical area and nutrition is not provided appropriately at that point in time, guess what happens? The cells in the cortical areas will be lesser than normal, will be malnourished, will be smaller, and of course the cells that have survived will die off. Remember, this is an almost permanent damage. It is an irreversible damage to the nervous system, to the growing nervous system. Why? Multiplying neurons require proteins. They require nerve growth factor. They require other trophic factors to move, to migrate, to develop connections. And if this process is blocked, this leads to a permanent deficit. So, by not feeding the mother appropriately, you are killing the next generation. You're starving the brains of the next generation. So, if you invest in feeding the mother, you're giving a healthy opportunity for the growth of the brain of the child. Imagine. So, what are the consequences? Let me go one by one. evidence, very strong evidence for a large number of variations that can take place. First of all is cell size. Cells do not develop, and there are, in fact, and if you look under the microscope in the starved brain, and they have done so with the rat brain, the rat brain of the starved rats, the brains of the starved rats, and the sections from the brains of the normal rats show that the starved brains have smaller cells, and let's call them the starved cells. The cell size is smaller. Of course, इसको खाने को मिला, इसको खाने को नहीं मिला. इसके पास वो न्यूट्रिशन ही नहीं है कि ये ग्रो कर सके, इसके पास थी. So what we have is that neurons are starved, the cell size is smaller, and this is, and we'll go along, और आप आगे भी देखिएगा. Therefore, अभी से आपको यहाँ से नजर आ रहा, जिस ब्रेन में सेल साइज छोटा होगा, सेल की प्रोलिफरेशन छोटी होगी, उस सेल में क्या होगा? वो सेल इंटेलिजेंट एफिशिएंट काम नहीं कर सकेगा. It will not be. So, this however is reversible if nutrition, proper nutrition is provided early. So, the cell can grow, but it will not grow to this size. It will grow, it will not grow to the normal size, but it will, from the starved state, it will come up to some level. So, there is possibility that you can increase the cell size. But what you cannot increase is the next thing, which is the cell number. And you do understand why the cell number will be affected, because cell division is necessarily requires each cell to use up a lot of energy. A lot of energy goes into each cell to subdivide and to multiply. And if the cell itself is starved, it will not multiply. So the cell division is affected by the malnutrition of cells. Therefore, proliferation will not take place. Cell proliferation will not take place. Because there is not enough nutrition, cells will not multiply. So, proliferation, multiplication, and therefore, number of cells will be less. If nutrition is provided during this period, when the cells are proliferating, of course, immediately cells will divide, and of course, the proliferation will take place. And of course, at that point in time, during that boundary, we can reverse that effect. But once that boundary is crossed, the time bound limit is crossed, we cannot reverse this. We cannot make the cells proliferate after the process has ended. So, no change is possible. And this, therefore, cell number, starved brains have fewer cells as compared to the nourished brains. So, cell size there is fewer smaller cells, but we can to some extent increase the size by providing some nutrition, but we cannot bring it to the normal level. Cell number, only if we provide the appropriate nutrition during the time that cells are dividing, the period, can we reverse it. Beyond that, it's lost forever. Cell number goes down and stays down in the starved brain as compared to the normal brain. So here is the second major effect. The third major effect is on cell connections. Remember, nerve growth factor, trophic factors, proteins, amino acids, growing, connecting, forming connections. Okay, so forming connections, it's not for free. This neuron is using energy and pushing its extensions and these extensions are made up of some substances, right? So, connections are being formed. Cells are sprouting extensions, dendrites, branches of dendrites, and the second order and the third order branches. Okay, so this needs nutrition. And if there is not enough nutrition, understandably, there is not enough of the dendrites. Now, you ask, what does it mean that the dendrites do not sprout? So what? The cell has existed. Cell is existing, right? जितने ज़्यादा आपकी टेलीफोन की एक्सचेंज की लाइंस होंगी और पॉसिबिलिटी होगी कि आप दूसरी एक्सटेंशंस तक पहुंच सकें, उतना एफिशिएंट सिस्टम होगा ना, राइट? और अगर वन लाइन से दूसरी लाइन से तीसरी, एक लाइन से दूसरे पे जाएंगे, दूसरे से तीसरे पे जाएंगे, तीसरे से चौथे पे जाएंगे, तो फिर तो देर लगेगी ना. लेकिन एक ही लाइन से अगर 5 डिफरेंट डायरेक्शंस में आप इंफॉर्मेशन पहुंचा सकेंगे तो वो एफिशिएंट है ना. योर कम्युनिकेशन इज एफिशिएंट. व्हाट हैपेंस इन दिस, दैट इफ द सेल कनेक्शंस आर नॉट स्प्राउटेड, you do have a neuron, but you have a neuron which is not very efficient. Now, which means having not enough nutrition, we have a decreased sprouting of dendrites which are bringing in information into the cell, and the spines or the branches of the dendrites will bring in more and more information for this cell to process and make meaningful decisions. Increased connections provide an efficient and a faster system of information. This is the same, you talk about information processing. This is exactly what each neuron is doing. So, the lesser information that comes into the cell, the lesser it will process, and the lesser it will process, the less efficient it is in terms of communication. And therefore, it is a starved brain. And therefore, a child whose brain is malnourished, extremely malnourished, will have a brain which is not capable of handling large amount of information, large amount of data. What are you doing? You're starving the brains of children by not providing nutrition to the mother. So, cell connections, cell number, cell connection, cell size. We have talked about these three. Next is myelination. Amazing, amazing. Myelination क्या है? Myelination है वो fatty covering, वो insulation. The formation of myelin, which is the covering for neuronal processes, like the covering for the insulation for wires, electrical wires, telephone wires, etc., etc. If nutrition is not provided in time, when the myelination is taking place, then cells are not myelinated. Inefficient system, again, where information is lost. जैसा electrical current लूज होता है, लीक होता जाता है और आप स्पार्किंग आपको नजर आती है. इसी तरह टेलीफोन की तारों में से आपको साउंड की जो लॉस है महसूस होती है. इसी तरह से जो न्यूरॉन है वो भी लूज करता है इंफॉर्मेशन बाय नॉट हैविंग माइलिनेशन. सो कैन इट बी रिवर्स्ड? वेल, नॉट एक्सटेंसिवली, बट कैन बी रिवर्स्ड बाय व्हाट वी कॉल न्यूट्रिशनल रिहैबिलिटेशन. समवॉट बट नॉट एक्सटेंसिवली. इसी तरह सेल कनेक्शन जो हैं वो भी रिवर्स हो सकते हैं लेकिन नॉट एक्सटेंसिवली. तो क्या है? आपने बाय starving the mother, you have starved the next generation. This is a horrendous thought, right? But then there are malnourishment of a very special kind, and we are going to talk about it. One of them is specifically only you provide them everything: carbohydrates, fats, but you do not provide the growing brain protein. Severe protein deficiency only, very specific. दूध नहीं देना और प्रोटीन्स नहीं देनी, माँ को नहीं देनी, और therefore the fetus, embryo and the neonate will not get it. In fact, you can guess, decreased neurons, number of neurons, decreased number of glial cells, decreased number of dendrites, decreased and deficient myelination, ineffective and inefficient system. Then this appears that the child, this disorder is known as marasmus. The child has irreversible brain damage. The decreases in the brain capacity is by 10 to 20%. The caloric intake goes down, the brain weight goes down. And so you see brain weights of the malnourished are lesser than normal and the brains are smaller than normal. They did this experiment with the malnourished rats. And then they took out the brains and measured them. On the scale, actually, you could see visibly that the malnourished rat brains weighed smaller, lesser, looked smaller as compared to the normal brains. Cell size थोड़ा है, cell number थोड़ा है, cell connections थोड़े हैं, cell के अंदर myelination थोड़ी है, तो फिर cell size थोड़ा होना ही है ना. And in some cases, this is an irreversible effect. Marasmus जो है वो selective caloric depletion है और Kwashiorkor एक disorder which is selective protein depletion, not deficiency, depletion. इसमें the baby has thin muscles, has fat and it has edema. इसमें vitamins भी आप दें लेकिन minerals, iron, potassium, calories ये सारी कम है. तो आप देखेंगे कि studies have shown, Monkeberg ने 1975 में 500 children of normal and 500 children, preschool children who were malnourished. Now astounding findings are that 40% of the malnourished children were below the IQ of 80. As psychologists and growing psychologists, you know, 80 में आप you're below the normal level. इसी तरह से उसने कंपेयर किया 500 normal well nourished बच्चों को and preschool बच्चों को, and he found out that 3% only out of these 500 had an IQ below 80. तो क्या नतीजा निकलता है? नतीजा ये निकलता है, it is in your hands to starve a brain by starving the mother and by starving the fetus thereby. Weiner ने 1977 में एक रिसर्च की थी. Mothers innately, especially in the animals, rats, know that this starved child needs or the baby needs more attention. And he found out that mothers spent more time with the starved or malnourished young rats than they did with their normal offspring. They know they need it. So maybe there is a lesson in there that we need to learn from the rats. Look after your young. So one thing that is now established is nutrition is extremely important. Nutrition is important for the development of the brain, and nutrition before or after the growing period is not going to reverse in most cases the effects of malnourishment. The second major influence that we are going to talk about today is hormones. Basically, if we have a list, we could divide it into the sex hormones, the male and the female, and the growth hormone, which is the thyroid, which is known as the growth hormone thyroid. Hormones affect the developing brain and the body. So it's not just the brain. You know, how does it affect the brain? It affects the brain by providing a template if it is sensitive, female hormone sensitized brain in the early period of development. During the later period, during puberty, the brain will respond to the female hormones. So the template and the sensitivity is laid during the early period of development. So hormones affect the developing brain and the body by the presence and the absence of the hormones, whether these hormones are present at the time or they were absent at the time. So the body and the brain grow accordingly, or increases and decreases in the normal level of the hormones that are required for the brain to be sensitized. If they are more than normal, we have a different response. If they are less than normal, we have a different response. The sex hormones are the androgens, the testosterone, and the female hormones, the estrogens. We have progesterone, estrogen, so on and so forth. So we're going to talk about this. Androgen, which is the male hormone, is extremely important for the developing and template laying of the neural substrates of male organs. It's not just with the double X or XY that the organs develop. It is with appropriate hormones at the time of development that this genetic material is then translated into the form. Okay, so these neural substrates of male organs and then male sexual behavior is laid down. So the template and the program is laid down by the appropriate male hormones. The release of these male hormones determines what reproductive organs will grow and will elaborate. But they also, not just that these organs will grow, elaborate and function, but also how the brain is organized so that it responds to the male hormones. If the prenatal hormones are absent, what will happen? The brain will not develop a template and will not then. Template development means the brain will trigger a response from the hypothalamus, will respond to the pituitary, will respond to the body, and the body will respond again. So the whole circuitry is then developed. Now, if the prenatal hormones are there, the organism would develop a hypothalamus, which would then direct the pituitary to release appropriate hormones, which would go to the target organs, which would then give an appropriate response. And if this is absent, then the brain would not be able to direct the hypothalamus, to direct the pituitary, to direct the organs, to release hormones to direct the organs. The pituitary releases gonadotropin, which is a hormone in a very tonic fashion, pulsating fashion, in a cyclic manner. In its absence, the pituitary cannot do this. It takes on the cyclic patterns of the females. So it releases the female cyclic pattern. So therefore, the pituitary is dependent upon the hormones that are released. Now what happens if female, the genotypically female, the double X female fetus is injected with male hormones? What do you think would happen? Of course, the male tonic pattern and the male template will develop. So these females will have more masculinized characteristics. And if they are injected with male hormones in puberty, they will act like males. These females who were genotypically female, injected with male hormones during early gestational period will be, as we call them, pseudo-hermaphrodites. And there are many cases in which such accidents have happened. These females will have male plus female organs, and transformation, there have been experiments done on rats, dogs, sheep, and a whole series of experiments have shown that if you inject male hormones in the prenatal period and then later on in puberty, you add or change the female behavior into male behaviors. The tomboyish females that you see are those who've had androgen exposure during early prenatal and postnatal period. That perhaps might also explain, and there have been hypotheses about it, the sexual preferences, the differences and difficulties of gender identity, such as transvestites and transsexuals. And we may not really hold these individuals responsible because this may be something which happened beyond their own control before they were born, the prenatal period when their mothers were exposed. So that is something that you need to understand, that hormones, especially the male hormones, can influence and can bring about a change in the total sexual identity physically of the fetus. The estrogen are not so important for bringing about drastic change in the nervous system or the body, but there is a controversy. The controversy is because there is evidence of feminization of the male fetus and the development of female organs in the male fetus upon repeated injections of estrogens. So you can see that the levels of hormones that are required for the genotypically male or female is important because the phenotypic characteristics do depend upon the appropriate level of hormones. The organism is male or female, whichever it is, if not stimulated with androgen, will emerge with a brain which responds only to estrogen and not to androgen, a typically female behavior and a typically female brain. And of course, there's a lot of controversy on that as well, but most studies do indicate that yes, there is evidence for androgen influencing or estrogen influencing the brain and thereby behavior. Thyroid. Thyroid is the third hormone that we will talk about. It is an extremely important hormone because it is a hormone which becomes increasingly important, especially around the last trimester, which is between the 6-month gestational period to the 9th month just before birth and then onwards. Thyroid hormone deficiency. If severe thyroid hormone deficiency, in some cases mothers do have thyroid deficiency and they pass on this deficiency to their young. And we have seen cretinism, which is a highly visible degeneration, and that results in a brain which is deficient. So thyroid hormone deficiency, especially during the last trimester, will result in a poorly developed cerebral cortex, will show that there is lower metabolic rate and there is lower glucose absorption in the brain. Lower glucose absorption means cells are not using that much energy. So cerebral cortex is starved of the substance of the hormone that it needs to grow and to sprout connections. So therefore, we see a cerebral cortex which is deficient. Decreases in brain size, decreases in the number of neurons, decreases in the number of axons, decreases in the number of dendrites, decreases in the connections between axons and dendrites, decreases also in the electrical activity of the cells. So the cells, the charge that you see in the cells, the electrical charge or electrophysiological activity is also decreased. And the good news is that this is a reversible effect. If thyroid is given, thyroid therapy, replacement therapy is given during the first year of life, then we can reverse it. If it is given early enough, when the presence of thyroid hormone is necessary or thyroxine is necessary for the brain, it can be reversed. But beyond that, it will not make a difference. Okay, so hypothyroidism is bad. Hypo means less amount of thyroid is bad. It is devastating. The brain will not grow. Is then the question, what about hyperthyroidism? Is it good? No, it's also not good. If having lower levels of thyroid is bad, having higher levels of thyroid must be good. Now, you need only the normal levels of. There is an early sprouting and early growth when the brain itself is not ready to make connections, the cerebral cortex. So we need to look at how keeping and maintaining the normal levels which the nature very effectively has balanced. Thyroid deficiency reversible only between the first year of life. Beyond that, no, it will not. And what we have is a brain which is deficient, and this is cretinism, a disorder which damages the brain. And once the brain is damaged and we go beyond to the first year and beyond the first year, this is an irreversible effect. Then there is another hormone, adrenocorticotropic hormone. Let me just call it ACTH. Where does ACTH come from? ACTH comes from each one of us. We secrete it when we are in stress. Adrenocorticotropic hormone is secreted by the adrenal gland for the fight and flight response. And where does the fight and flight response come in? The fight and the flight response in nature is readying you for battle. Evolutionary history को याद करें तो ये क्या है? It is a response, और खास तौर पे अब हम इसको कहते हैं the stress response. Is stress good for the growing fetus? How does the fetus get this stress? पहली बात through the mother. दूसरी बात through the loud sounds and noises that are around it. Constant stress. लीवाइन ने ये बहुत 1960s में रिसर्च हुई थी और बड़ी इंटरेस्टिंग रिसर्च है, और उसने ये रैट्स पे रिसर्च की थी और उसने कहा कि और कुछ ना करें रैट्स को, ये आप बेबीज के साथ भी करके देखें, सिर्फ उनको उठाएं, हाथ में हैंडल करें और उसके बाद दूसरी जगह आप डाल दें. एंड दिस इज कॉल्ड हैंडलिंग. लीवाई very interestingly said that mild stress is good. Mild stress of handling a puppy, handling a baby is good. Handling और mild shock, उन्हें mild shock भी दे के देखा, छोटा सा shock, not very strong, and that also brought in a stress response. Handling also brought in a stress response, which then released adrenocorticotropic hormone ACTH from the adrenal gland. And what it did was showed, very interestingly, he tried them out in different experimental situations. He found out that the handled rats had matured earlier. So precocial development है. If they were given small, mild stressors, they matured earlier to adulthood. And if they were placed in a strange environment, a novel environment, they were not afraid. They would explore, go exploring the corners. Normal, unhandled rats, pups, would defecate, urinate, or squeal, and they would be afraid in that environment. But not these handled rats. So they were active, running around all over the place. So they were compared to the undisturbed, which defecated, which matured later, which could not cope with the stress of a novel situation. These rats did very well. So, handling increased ACTH, increased adrenal secretion, and it increased a cycle of quicker absorption. So, early development of stress response is therefore found in these animals. Usually in the young rats, ACTH is released around 16 days of age, but in the stressed rat, it appeared 4 days earlier. Similarly, for the humans. The lesson is, maybe handling babies, holding babies is good for them. And letting them explore after you've handled them is of course going to make their brains and their responses much better. But too much stress can be bad as well. Why? Why do you think too much stress can be bad? Too much stress can be bad because research has shown that if we increase the intensity of the stress, mild stress is good, but increase the intensity of the stress, and research was done by Ackerman, Hofer and Weiner in 1978, and they compared adult rats, grown-up rats, which were separated from the mothers at 15 days, and rats which were separated, adult rats, which were separated from the mother at 22 days. Now, I must tell you that rats are weaned away from the mother at about 21 days. So, before they were ready to separate from the mother, and were a group of rats which were separated at the normal time of weaning. And lo and behold, what they report is the early removal from the mother leads to a high risk of ulcers, which is ulcer is a stress response. You do know that. Those who are in high stress jobs develop ulcers. We know that. And in the rats, we also see that those rats which were removed from the mothers at an earlier age had a response which was similar to the stress response. But not just this, they were also deficient in a large number of behavioral variables, biological and behavioral variables. They also had defective thermoregulation. They could not control their temperature, whether if it was a cold challenge or a hot challenge, they could not regulate the body temperature. So these were rats which had a serious defect in their normal growth. So what do we see here? We have talked about influences, and we have talked about and we have seen how dangerous it is to change the normal levels of hormones, normal level of nutrition, normal level of substances, and we'll go into a lot more of these, and you'll be surprised how careless we are when we are dealing with the next generation. Experiments have shown that it is a very sensitive matter, growing within the mother and growing outside the mother. We'll talk about it in the next class. Do read Pinel, but also read the handouts and also go over the PowerPoints that you might have. Very interesting, and I would like you to also make observations.